and thought, they just kind of sit there. They don't do anything very interesting. They don't behave. They're not responsive. They don't sense their environment. Things come along and just eat them, and there's not much they can do about it. If you're thinking any of those things, I hope that by the end of my 45 or 50 minutes that I will have changed your mind about all of those things. So today I'm going to focus on plant sensing and communication. And before I start, nowadays science isn't done by yourself. So I'd like to thank my main collaborators, Will Wetzel, who's now at Cornell, Kaori Shiojiri, who was a postdoc with me about 12 years ago, and we've been continuing to do this work ever since. Satomi Ishizaki, who's also from Japan, she's a geneticist, and James Bland, who's a chemist. He's English, but he's at the University of Finland. And I also felt kind of sorry for James. He didn't have a hat, so I gave him one there. Okay, so I'm going to structure my talk today around these 5 questions. What is communication? Do plants communicate about herbivory? Why might plants respond to cues? Why might plants emit those cues? And then who communicates in a little bit about how do they do it. So let's start with the first one. What is communication? And I'll tell you, this is how I define communication. And if you don't like my definition, I'm fine with that. I don't think this is the only definition, the best definition, whatever. But this is how I'm defining it. So let's put it out there right away. And I think that if you don't like this definition, we can talk about it later. Please don't. Sometimes people get stuck here and they think, well, I don't think that definition's right. And then they have a hard time listening to the rest of the talk. So I'm not especially committed to this definition, but this is the one that I use. And that is that communication has occurred when one individual, one plant, emits a cue. And in my case, I'll be talking today about volatile cues. And that cue is perceived by another individual. And as a result, the receiver changes in some way. And these changes are conditional and they are short-term. They're reversible. Okay, so this is what I mean by communication. This transfer of an information-rich cue. And let's move on and ask, well, do plants do this? Do plants communicate about herbivory? And this isn't an especially new question. People have been presenting evidence that this can occur since when I was a graduate student. But this has been a field that's been really controversial. And it's not entirely clear to me why it has been so controversial. We know, for instance, that plants respond to a lot of things in their environment, a lot of environmental information. So, for instance, if plants get shaded, they respond to light cues, and they change the way that they grow. Plants that get shaded grow a lot taller. We know, anybody who's ever had a house plant knows that plants will grow towards the light, towards the window. We know that plants can perceive information about light and respond to that information. Similarly, we know that plant roots are able to sense the availability of water and nutrients in the soil. In this particular experiment, and this is quite old work from 1975, these barley plants were grown either in an environment that was homogeneous, that was rich throughout in terms of nutrients, and that's the case for this plant on the left. The plant on the right was grown in soil that only was rich in nutrients, through that one band through the middle there. As a result, the plant only proliferated roots in that narrow band where there were lots of nutrients. So the plant was able to perceive its environment and respond accordingly. So it's not clear to me sort of what all the fuss is about, why plants communicating about herbivores is really all that different from some of these other examples that are, I think, much better accepted and have been for a long time. But I'm in a department of entomology, so we're interested in bugs and in how feeding by insects affects plants. And it's a wonder to me that I got hired in that department, that they kept me. Nonetheless, I have felt some obligation to focus my work at least a little bit on what happens when plants get eaten. Are they aware of that? Do they change accordingly? And do they communicate about, are they perceptive about what's going on to their neighbors in terms of, are their neighbors getting eaten by herbivores? And so the system that I've worked with in asking whether or not plants communicate about herbivory, which is my next question. The system that I've worked with since about the early 90s is sagebrush that grows on the east side of the Sierras and through the Great Basin in North America. It's a very common plant in this habitat. And the experiment that I'll start off by telling you about involves pairs of sagebrush plants out in the field. So I've got pairs of plants, and one of the plants of that pair I can experimentally clip. And so that is the treatment that's shown there in the upper right. There are two sagebrush plants, and in this case with scissors, I come along and I clip the leaves of one branch of one of those plants, the one that has that pair of scissors near it. And the plant that is the assay that's within 20 centimeters of the clip plant does not get any clipping itself, but over that season, I record how much natural herbivore damage that assay plant receives. So that's my clip treatment. And then I've got a clipped and bag treatment. I do the same thing. I cut the plant with scissors, but in this case, I put a plastic bag around the clip branch immediately after I scissor it. And this reduces or eliminates air contact between the clip branch and the assay branch. And here, I also record over the growing season how much damage the assay branch receives. So that's my clipped and bag treatment. Very little air contact between these two. And then I've got on the bottom a control where I don't do anything to the unclipped branch here, but I record how much damage the assay gets. So my clipped branch, there's air contact. In my clipped and bagged, limited air contact. And in my control, there's just nothing going on. And what I find when I do this is that, and what I'm measuring here is the amount of damage caused by herbivores to the assay branch. And what I find is that for the assay branch that's right next to a clip branch, it experiences less damage by herbivores throughout the growing season relative to the branches near the clipped and bagged branch or the control that was never clipped. So something about being near a clipped branch makes the assay branch increase its level of resistance so it gets damaged less. That's kind of curious. And we've repeated this experiment a bunch of times. The first paper, we kind of repeated it a couple seasons, sent it into the journal, got rejected, tried a few more journals, got rejected. So we wound up that experiment repeating it for 5 years before finally we were able to sneak it in. And so we've repeated this now very many times in a bunch of different places. And so we've learned a lot about how this process works with communication between sagebrush. I should point out that sagebrush often has another sagebrush plant as its nearest neighbor. So this is something that can occur in the field. We can find evidence for communication when the plants are separated by up to about 60 centimeters. So about yay far apart. And almost all of the plants in the Great Basin, almost all sagebrush plants in the Great Basin, have neighboring sagebrush plants that are within 60 centimeters. So this is something that makes some ecological sense that could possibly happen in the real world. We find that young plants are both the best emitters and receivers. And in the experiments that I described to you, we did the experimental clipping with scissors because that's easy. But we were also concerned, we also wondered whether or not scissors were indicative of real herbivory. So we have done similar experiments by doing the clipping, bagging either grasshoppers or beetles onto some of the leaves, allowing them to do the damage. And we find much the same results as when we do the clipping with scissors. So now we just use scissors because it's a lot less of a hassle. Okay, so this is a result that we can get repeatedly. Well, is sagebrush some weird plant that's out there? This is a meta-analysis. So this is a table that I don't expect you to be able to read, but each of the lines, each of the rows is a different plant species, a different study. And for those studies where the communication resulted in the neighboring plant becoming more resistant, those are shown as the effect being to the left of this vertical line. For those studies, and there were some, where the neighbors actually became better hosts for their herbivores after having their neighbors be attacked. Those are to the right of this line. The early studies were criticized for not being very well replicated, and those are shown, those are the studies at the top, and they have no error bar because they were not rigorously replicated. And I think early skeptics were critical of these studies because they weren't carefully replicated. But I think that now that these experiments have been done more rigorously on a lot of different species, I think you can see that many species show this kind of communication that results in greater levels of resistance without them actually being damaged, just if their neighbors have been damaged. And so if we sort of tally this whole thing up, the overall effect size is the last little circle shown at the bottom, and it is an effect that is significant and in the direction of greater resistance. So this kind of phenomenon is not just some weirdness of sagebrush, but in fact something that we see in a lot of other species. Okay, so why might plants respond to these cues? And this isn't especially troubling. It makes sense that plants will be under selection just like animals are, and that there are risks for plants, and plants that are sensitive to cues in the environment are able to adjust their defenses, adjust their traits, so that they are better suited to growing where they grow and have higher fitness. Well, this all sounds good. Is there any evidence for this kind of thing, for plants that respond to cues having higher fitness? Again, using the sagebrush system, which for lots of questions is really a very poor choice, but it is in a nice place, and it is a system that I know. So we have continued working on it, and one thing that, one advantage to using sagebrush, or one of the things that we wondered about is whether or not this kind of communication was required for a plant to coordinate its own systemic defenses. So what I mean by that is that if we get an infection in one part of our body, if I get an infection on my left index finger here, I recognize that infection, I have an immunological response, I produce white blood cells and who knows what else, and these cells move throughout my body so that systemically I am geared up to be more resistant to subsequent attacks. So I am capable of systemic resistance throughout my body, through my vascular system. Well, it turns out that plants can do this to some extent, but their ability to move cues around their vascular system is not very good. They're most able to do this when they're growing rapidly, when water is not limiting, and plants like sagebrush that grow in arid places are very poor at this kind of vascular communication within an individual. So it turns out that for sagebrush to have systemic resistance, to mount systemic resistance within a single individual, volatile communication from one tissue to another, from one branch to another, within that individual is required. And we know that by doing an experiment that's very similar to one that I showed you earlier with pairs of plants, except here we're just looking at branches, different branches within the same individual plant. So in the upper left here, I have a clipped plant where I have clipped the leaves, again using scissors, on one branch, and I'm assaying what happens on a neighboring branch of the same plant. So here, there's the opportunity for vascular communication because the branches are connected. So cues can move through the vascular system of the plant, and also for volatile communication. In my second treatment, in the upper right, it's the clipped and bag treatment. So after I do the clipping, I enclose the clipped branch in a plastic bag. So here, there's the opportunity for vascular communication, because again, the branches are connected, so cues and signals can move through the vascular system, but the opportunity for volatile communication through the air is greatly reduced because I've bagged the branch. And then I've got my control where I don't do anything. And again, what you can see here, when I assay, when I measure the amount of damage that the assay branch receives from natural herbivores throughout the growing season, that where volatile cues were allowed, only in my clip treatment, that's the only treatment where I see a reduction in the amount of damage that the assay branch experienced. Where vascular communication was allowed in the clipped and bag treatment, but not volatile communication, damage levels are high, and for the control, damage levels are high. So for a plant to coordinate its own defenses, it requires these volatile cues. So that's one advantage. Well, does this translate into changes in the kinds of fitness measures that we can get for sagebrush? And as I mentioned, sagebrush is long-lived, slow-growing. It's a pain in the butt to try to propagate. So it's not a very good system to ask these questions. And in most years, we don't see a lot of seedlings. We only see a lot of germination in years when it is really rainy and the rains go late into the year. And these 2 years, 2010 and 2011, were both really wet and late wet years. So there was lots and lots of sagebrush germination. The floor kind of looked like this in a lot of places. It was just kind of a carpet of little sagebrush seedlings. That was exciting to me. And I decided I needed to drop everything and follow what happens to these seedlings. So in both of these years, I started with over 1,000 seedlings. And for half of those seedlings, I clipped their neighbors, the adult neighbors that were near those seedlings. And for the other half, I didn't do anything. And then I followed what happened to those seedlings over the course of this first season until the end of the rains came in early October that year. So the dry season was finally over. And what you can see is that most of the seedlings died, but that in both of these 2 years, the survival of seedlings was about 10 times as great for those seedlings that were near experimentally clipped neighbors compared to neighbors that weren't clipped. Similarly, I have followed a cohort of established plants for 13 years, and in that 13-year span, I experimentally clipped their neighbors 5 times. And plants that were near experimentally, or branches that were near experimentally clipped plants produced more new branches and more new inflorescences, more new flowers than branches that were near unclipped neighbors. So we're still a far way from really getting a handle on sagebrush fitness, but this just is not a good system to ask questions about fitness. And to the extent that we're able to estimate fitness, and really this is something that most of the other people who are doing this work have not asked about the fitness consequences. This is as good as it's going to get. So why might plants respond to cues? It allows them to coordinate their own systemic defenses, and it also seems to translate into greater survival of seedlings and growth and reproduction of established branches. And I guess that's not too surprising, but these plants are competing with one another. They're competing for light. They're especially competing for water. If these cues make my neighbor, if I'm damaged and emitting these cues makes my neighbor do better, why in the world would I want to emit these cues? Why am I helping my neighbors? This seemed a lot harder to explain. It was a lot more kind of troubling. And so I've considered, I think, 6 or 7 alternative hypotheses for why plants might emit these cues, and I'll run through these for you. The first one is that they leak out. They're not adaptive. So I'm a bit of a raving adaptationist, and so I want to see a benefit for all of these traits. But it might be like asking, why do we give these highly visible red cues to our neighbors when we cut ourselves? And I think that that's a kind of goofy way of looking at it. We're not giving a red cue. We're bleeding. We can't help it. We're not attempting to communicate. the cue is not adaptive. It just leaks out. It happens. And so maybe that's what's going on here, that there's no selective benefit by providing these cues. And I don't know if that's the case. I don't really know how to address that issue. But I think it's worth mentioning because it's possible that that's what this is about. A second possibility is that these chemicals that get emitted when plants get damaged repel herbivores. And there's evidence from other systems that this kind of thing takes place. So we were interested in seeing whether or not when sagebrush is damaged, does it repel the herbivores of sagebrush? And we were not able to find any evidence that this was the case. So we'll say no, or at least we couldn't find any evidence. A third possibility is that these chemicals attract mutualist predators. And this has also been found for a lot of other systems. When plants are attacked, they emit volatiles, and predators and parasites of their herbivores are able to sense these volatiles, sense these cues, and key in on the herbivores. So it seemed possible, although I guess people always ask me that at the end of seminars, and I never really liked the idea. It seemed like, come on, the herbivores here are things like grasshoppers. They're moving around all the time, and jackrabbits and deer. Are the plants really going to call in the predators and parasites of these mobile and big herbivores? It seemed very unlikely to me. Nonetheless, everybody wanted to know this, so okay, I'll take a look. Do these chemicals attract the predators of the herbivores and therefore maybe help the plants? And the herbivores that were abundant in this system were predaceous bugs and predaceous ladybug beetles. And much to my surprise, when I set up sort of a trap line of plants that I either experimentally clipped or didn't clipped, and then just recorded how many predators are attracted, how many predators do I see on those plants when I visit them every day. Much to my surprise, so this is the number of predators on each plant. And what you can see is that, well, let's start with the control. Most of the control plants didn't have any predators visiting them. A few of them had a few predators. Plants that I experimentally clipped, much to my amazement really, attracted more predators. So it did seem as if these chemicals were attracting the predators of herbivores. However, in this system, in the sagebrush system, unlike other examples, spider mites on beans, these predators didn't reduce levels of herbivory on sagebrush. So these predators weren't making much of a difference in my system. So I will say they attract predators. Well, yeah, they do attract predators, but no, those predators don't make a difference. I don't think that this is driving the evolution of this communication. They might inhibit the germination of competitors. So some of the chemicals that we knew were emitted by damaged plants were also known to be pretty powerful germination inhibitors. So we conducted some experiments, first out in the field, where sagebrush and most of the plants in this system germinate in the late spring, in May, as the snow is melting. And so the window of germination here is pretty short. And so what we did, right as the snow was melting, we experimentally clipped some of the leaves of sagebrush, and then we had others that were controls where we didn't experimentally clip them. And then we recorded adjacent to those clipped or unclipped branches, how much germination did we see? And what we found was that there wasn't much germination of sagebrush seedlings. There was a lot more germination of just volunteer dicots, things that came up. And what we found was that adjacent to plants that we had clipped, there was much less germination than adjacent to plants that we didn't clip. Ooh, this seemed kind of interesting. So we thought, okay, we can do this in a lath house. So a lath house is just a bunch of benches. And instead of being inside like a glass house, a greenhouse, it's inside a house that has a little bit of wooden, little wooden sticks that give it some shade, but not a whole lot. So it's basically outdoors with a little bit of shade. So we did these experiments back in Davis in a lath house, and we brought soil back from our field site and put it in pots. Into each of those pots, we planted one sagebrush plant. We either clipped that plant or we didn't clip that plant. And then we also planted a few sagebrush seeds and a few wild rye seeds. Wild rye is a grass that grows here. And what you can see is that there was no difference in the germination of the sagebrush seeds, whether or not the plant had been clipped. Wild rye germination was reduced in this clip treatment relative to the control. But again, we only put in a couple seeds. Most of what we got in our pots were volunteers, seeds that were there naturally. And the germination of volunteer dicots was much reduced where we clipped the sagebrush, as was the case for volunteer grasses. They also didn't germinate nearly as much when the sagebrush has been clipped. So it does seem as if these chemicals that are emitted when sagebrush gets damaged are pretty good inhibitors of germination. So this seems to be, yes, something that might be a consequence of emitting these cues. They might allow systemic resistance. This is a slide I've already shown you, and indeed that's the case, that when volatile contact, when there's airflow between a damaged branch and an undamaged branch of the same plant is possible, the plant can coordinate its own systemic defenses. Without that airflow, it's not able to do that. And that could favor the emission of cues. In other words, communication between plants might be almost a secondary effect of communication within an individual plant. And that seems like a possibility. And the last possibility is that this communication may allow kin selection. So let me explain this in a little bit more detail and then spend a few minutes telling you about the experiments that we've been doing more recently to look at this. So we found that communication was effective and was required for a single individual to coordinate its own defenses. And that got us thinking that maybe that's really the driver here. And if that's the case, we wondered if plants would be able to distinguish between their own cues versus the cues of another plant. Can they communicate? Will they respond more effectively to their own cues than to somebody else's cues? And so in order to test this, we tried to propagate sagebrush. This got us, I forgot I had this slide in here, but this got us sort of going down a path of trying to understand who communicates and how they do it. Here's the experiment that we did. Sorry, I got a little bit out of order here. Here's the first experiment that we did that was an attempt to ask whether or not sagebrush is able to communicate more effectively with itself than with another plant. We were able to, after several years of trying and not getting very far, we were able to take cuttings to essentially propagate or clone sagebrush individuals. So that we started with plants out in the field, such as this plant on the left, rooted out in the field, and we were able to take cuttings of it in the autumn, bring them back to Davis, get them to root and produce new plants that are genetically identical to the mom. And then we brought those plants back out to the field in pots and put the pots next to either the individual that is genetically identical to it, and we're calling that self, or to a genetically different individual that we're calling non-self. And then we can clip the potted plant, the either genetically identical self plant or genetically different non-self plant, and see what effect that has on the rooted plant that we started with. And so we did this, and we also had a couple of other controls that I'll explain to you. And this is a little bit of a complicated slide. So here we've got 4 treatments. We've got an unclipped control plant. That's pretty intuitive. We didn't do anything to it. And it got a lot of damage, as you can see. We have a treatment where we actually damaged some of the leaves of the assay plant. And that got very little damage. And both of those, in a sense, both of those are controls, because they kind of set the bounds for us. The unclipped control shows us, ooh, how much damage will a plant get if it's not receiving any cue. And the treatment where we clipped the plant itself is sort of a lower bound. How little damage will a plant get if we clip that plant itself? And then the other 2 treatments are the ones that we're really interested in, the neighbor clipped ones, where that neighbor was either self, genetically identical, or non-self, genetically different. And what you can see is that communication was more effective when it was occurring between genetically identical plant tissue. So plants are better able to communicate with themselves than they are with somebody else. That's kind of cool. Again, I think many people assume that this kind of ability to distinguish self from non-self might only be found in animals, but that's not the case. So at this point, we really wanted to know whether or not plants can distinguish relatives from strangers. And so there are techniques to evaluate how closely related individuals are using microsatellites. And I know that word, but I have no idea what this really means. But we collaborated with Satomi Ishizaki, and she was able to work up a system of microsatellites for these sagebrush plants and come out to our field site where we have this meadow of sagebrush and various pairs of plants. And she took samples and did whatever she does back in the lab in Japan, does her magic, and can tell us the amount of genetic relatedness between any pairs of plants. On average, how many of their genes do they share in common? And so we now have this meadow, and we know who's related to whom based on Satomi's magic. So we have, in the first year, we were able to clone some of these plants, put them in pots, and move them around. And so we have the Q coming from plants that are either close relatives or distant relatives of our assay plant. And when we did this, we found that in most instances, plants that were receiving cues from close relatives were less damaged than plants that were receiving their cues from distant relatives. So that communication was more effective between close relatives than it was between strangers. Well, the sample size here is kind of pitiful. It's about 20 individuals, and it was a lot of work to get this, just because propagating these plants is so difficult. But one of the things that we thought is that, instead of cloning these plants and trying to move them around, maybe we can just move air around. And so the first year that I did this, I did the clipping, collected the air inside of a plastic bag, sucked the air up, and then holding my breath, I walked over to other plants and exhaled the air into a plastic bag surrounding the other plant. And what I like about this a lot is that anybody can do this. So that, you can do this for a science fair project. You can, check this out for any plant that you happen to be near. Just clip it, collect the air in a plastic bag, hold your breath, walk that air over to other plants, and exhale on it. And sure, there are things that there will be interactions between, what's ever going on in your respiratory system in that air, but those interactions will be the same for in each of the cases. Well, reviewers really didn't think this was so great, and a few people also pointed out to me that at least for sagebrush, many of the chemicals that sagebrush produces are really pretty major carcinogens. So that this was probably a really stupid thing to be doing. So, as a result, we decided we can get the super syringe. So this is a 1-liter giant syringe that does the same thing, and you have a lot more time to walk between much more distant plants when the air's in the super syringe. And this allowed us to greatly increase our sample size. And so we essentially did the same thing. We moved air around from damaged plants to undamaged plants that were either close relatives or distant relatives. And when we did this, again, we found that when the cue came from a close relative, it was much more effective at inducing resistance and reducing damage than when it came from a distant relative. So communication seems like it's mostly with self-individuals or with kin. And this has a number of important consequences. It allows systemic coordination. It's unlikely then that an emitter will aid strangers. So I think I mentioned that one of the things that bugged me about this is why would a plant be helping its competitors? And in fact, it might not be helping very many of its competitors. If it is mostly helping its relatives, that makes a lot more sense. That's a lot easier to understand. It's also likely that plants are responding to cues from their kin. For instance, I'm much more interested in knowing my dad's medical history than I am in knowing, the average white male's medical history, because we share susceptibilities. And we are interested in knowing whether or not sagebrush individuals that are related also share susceptibilities, and that's something that we're hoping to get at. This is something that we have come across pretty recently. Each of these pies represents a single plant in our meadow. And the pies are just kind of thrown up there on an Excel spreadsheet, on an Excel graph page, whatever that is. But the position of the pie is not the plant's position in the meadow. And the colors in the pie show different chemicals that we were able to measure, or really that James Bland and Carrie Shiojiri were able to measure, because this is also something that I don't really know how to do very well. What they found was that in this meadow, kind of the plants are all interspersed here, that some of the plants, these ones that are shown as pies that are mostly orange, they are producing a lot of this one chemical thujone. Then there are other plants in the meadow mixed in with these thujone plants that are shown by this, I don't know what color that is, let's call it plum. And they are producing mostly camphor. Camphor plants show no measurable production of thujone. Thujone plants are, everybody produces a little bit of camphor, but not nearly as much as these camphor plants. And then there are a few plants scattered in the meadow, the ones that have these green highlighter around them. Oh, there's one up there. It's mostly light blue. And then there are a couple here that, unfortunately, the colors are kind of close. They're like this one. They're sort of burgundy. And they're just oddballs. They are producing other chemicals that aren't necessarily found. So there are plants of these different types. That same species, same subspecies, just out in these meadows. And they are producing, these different plants are producing really different volatile profiles, very different chemicals. What's this about? We don't know. But it seems like, this is pretty weird. Maybe a little bit like human blood types. And by the way, we don't have a very good handle after knowing about human blood types for decades, I don't know, centuries, and having, a whole bunch of people look at that problem with a whole bunch of research money. We don't have a very good handle on what these human blood types are about. And we certainly don't have a good handle on what these chemotypes are about in sagebrush. But one of the things that we found is that plants communicate more effectively with other individuals of the same chemotype than they do with other individuals of different chemotypes. So I don't know, maybe they are using chemotype as an initial screen of relatedness. We've looked at the chemotypes of parents and the chemotypes of the offspring of those parents and found that they are generally the same, so that chemotype is highly heritable. And what else is different about these chemotypes? Again, we've looked at a whole bunch of different traits. Plant, leaf shape, leaf size, plant growth form, morphology, leaf color, neighbors. None of those seemed like they were different. One of the chemotypes, camphor, was more common in the sun, thujone more common in the shade. That was last year. It didn't seem to, we looked at a bunch of meadows this summer, and that didn't seem to be the case. So we don't know what's different, but the herbivores and pathogens can certainly tell them apart. So something that we're not seeing, but they are sensing, is consistently different about these chemotypes, and this is something else that we'd like to follow up. So let me stop my talk and summarize what I hope you will take out of this talk, and that is that communication happens between plants. But this process is not random, and in fact, it's most effective amongst genetically identical tissues, amongst genetically related kin, and amongst similar chemotypes. So I will stop there, and I'd be glad to answer any questions that you might have. Thanks a lot. Any questions for me?
Yeah.
Yes, sorry.
Did you say that chemotype is genetic, as in the offspring of one plant are the same, form relatively the same phenotype as their parent?
Yes, that's right. So that it's highly heritable. So we're able to compare the chemotype of the mom and the chemotype of her offspring. And the offspring are not necessarily the same as the mom, but they are much more similar to the mom than you'd expect at random. So you can calculate the likelihood or the fraction of genes that are transferred from the mom to the offspring, and that estimate is quite high for these traits. Yes?
I don't know if you covered this, but what did you use to measure damage?
Oh, yeah, I didn't mention that. I just measured how many leaves got eaten by chewing insects. So I go back at the end of the season, and how many leaves are there, how many of them have holes in them, or pieces cut off the edges.
Was it a percentage of how many were there to begin with?
Yeah.
Have
Yes?
you looked at pollinator responses to chemotype?
I haven't looked at pollinator responses. I've just been, sort of onto the chemotypes pretty recently. Sagebrush produces really small flowers that are pretty inconspicuous. They are thought to be wind pollinated, but I think that I've become ever more skeptical of hearing that plants are wind pollinated. Oak trees are wind pollinated, but and we've got lots of valley oaks around Davis. There are sure a lot of insects that visit oak flowers. So I think the easy answer is, these things are wind pollinated. Nah, doesn't matter to pollinators, but I'm not so sure. So that would be a cool thing to take a look at, but we haven't done that. Yes?
Has this approach been extended to marine environments?
Yeah. So another system that there's been lots of good work done looking at communication is wild lima beans that grow in tropical Mexico. And at least in that case, sort of offhand, that's the one I can think of. Martin Heil has done that work, and he's got really nice evidence for communication. With those, the wild lima beans grow as vines, and it's very difficult to tell an individual, so that it's unclear there, who's connected to whom, what leaves are connected to other leaves. But he definitely has good evidence for communication between leaves that are on different vines. Are those different individuals? I think that's a little bit less clear. Yes?
I think this is more of a historical question, but in this field, it seems from the very first, when it broke, the talking trees tech moniker sort of thing, the emphasis has been on the adaptive significance of the plant communication from the sender's point of view as opposed to the receiver's point of view. I mean, a lot of your work shows that maybe the adaptation doesn't have to do with who you're communicating with, it's who you're eavesdropping on.
Yeah.
Okay? And that the whole thing, especially now that you've gotten to the genetics of this, is they simply become good listeners. If you're a wildebeest and a bunch of wildebeest being chased by lions like this, it's not necessarily beneficial for me to let you know that lion is there, but it's beneficial to watch you run.
Yeah, I agree with that completely.
Did the popular press maybe jump on it in a way that...
Yeah, I think that's the case, that it's pretty unfortunate that the popular press got pretty carried away, and that maybe that set the whole field back a decade or so. The Talking Trees moniker, I agree, is I don't like that as a shorthand. Like, I think listening trees, if you have to do that, call them listening trees instead of talking trees. Yeah.
because I've always thought your clipping response within the plant really points to that.
Yeah, I agree that it's much easier to understand why selection would favor a plant being sensitive to and responding to cues in its environment of any sort, light cues or these chemical cues, and that makes a lot of sense. Whether or not it is adaptive for a plant to be giving out this information is a lot less clear.
Is the chemistry such that it would make sense that the siblings would be more responsive to the chemical produced by self or whatever, just because how the plant responds to the chemistry to that?
I'd say we don't really know. We can measure, better at this point, better than 100 chemical peaks, and which of those, are any of those, the active stuff is really a difficult question. We've taken a sort of pharmacological approach, and you can buy most of these chemicals, from Fisher, get them in a little bottle, put them on a wick, put them out near these plants, and at those concentrations, and many of these chemicals are very biologically active. So we can find that plants respond to these chemicals, but is that what actually happens in nature? And are those concentrations at all realistic? And then it becomes like way more difficult to answer those questions. Yes?
How did you settle on sagebrush? Was it just because it was accessible?
This is a long story that sort of has nothing to do with science, but everything to do with sort of my kids, and maybe I reviewed a paper years earlier by these biochemists Bud Ryan and Ted Farmer, and they had done an experiment in a vacuum jar where they found that clipped sagebrush, which is a mega producer of this chemical methyl jasmonate, a plant hormone, but they didn't know at the time. This is one of the really key experiments that showed that methyl jasmonate was a plant hormone. So they did this experiment where they incubated methyl jasmonate with potted tomato plants and found that in a vacuum jar, and found that the potted tomato plants produced all these really cool chemicals that were defensive. And so I was the reviewer for this, and I thought, whoa, this is convincing, it's a good experiment, but it's also like really weird. Sagebrush plants, potted tomato plants in a vacuum jar, like these plants never encounter each other in nature. Like, what is this weirdness, and what are we doing with this? This is unnatural, but it is cool. Okay, let's go with it. And then I happened to be in this place because it was a good place for my kids to be. I was working on wild tobacco because I was friends with someone who worked on wild tobacco and the chemistry of tobacco because people have liked to smoke it, and there was a huge industry associated with it. So the chemistry of wild tobacco was quite well known, and a lot was known about tobacco. And so I'm kind of working on wild tobacco because I want to spend my summers outdoors. And I kept stepping on sagebrush and thought, wait a minute, I read something about that. And sagebrush and tobacco and tomato are pretty closely related. So I thought, maybe that weird paper that I read actually has a natural analog. And so that is the short answer, if you can believe that, to how I came on this. Yes?
Really enjoyed your presentation, and there's lots of thorough work here, and it had me thinking. Everything you mentioned, when these signals are emitted, they sound like a benefit both to the plant that was damaged and to the neighbor, and kin or not to a greater or lesser degree. So my question is, there must be a cost somewhere to sending out these signals. Otherwise, what would keep a plant from just always broadcasting these signals all the time?
Yeah, I agree. I think there have to be these kinds of costs. I mean, why do the plants wait till they get these signals to turning on their defenses? Why not be maximally defended all the time? And we know a little bit about this. We know that when plants are, for many plants, when they are well defended against insects, they are poorly defended against pathogens, for instance. And that's, there's a fair bit of evidence for that. In the field, when people have looked for those kinds of costs, they have been very hard to find. And I think that part of that is that the expectation is a little bit, maybe is a little bit naive. So that, and I'm someone who's done some of these experiments, and my expectation is that defense is part of a zero-sum game. So I don't know if you guys have heard this terminology. It's a little bit of jargon, but basically the idea here is that we're assuming that a plant can do kind of two different things. It can either defend itself, it can invest, it can allocate resources to defense, or if it doesn't allocate resources to defense, it can allocate them to fitness, let's say, to reproduction, to growth and reproduction. And I think that that expectation is a little bit naive in the sense that a plant can do so many different things, so that I've set up experiments looking for that kind of a cost. If I get a plant to invest more in defense, oh, then I'll be able to see that it has less for fitness. Well, a plant, if it's not investing in defense against my particular herbivore, it can defend against pathogens, it can invest in roots, it can, do a gazillion things, and I'm not measuring all of those. So finding the empirical evidence for those trade-offs, for those costs, has been very difficult. But I quite agree. They've got to be out there. It doesn't make sense if they're not. But, they're hard to find. Yes?